Good afternoon. I'm Jim Baker. I'm Head of QARA and Clinical Affairs at Renishaw Neuro Solutions, and we manufacture the Neurofuse drug delivery system. And I'm going to present some aspects of the data coming from the trial from the device point of view. Wait for the presentation to come up. There we go. Okay, so next slide, please, Rebecca. So the drug delivery device is implanted using a technique called stereotactic surgery, which I talked through the video earlier. So using this technique, the brain and skull are essentially mapped using Renishaw NeuroInspire software, seen here at the bottom left-hand side of the slide. And medical imaging, such as CT or MRI, and a target in the brain is defined from that. NeuroInspire then talks to the robot. The robot is a stereotactic robot, also manufactured by Renishaw and seen here on the right. And this accurately implants the catheters and port. After the implantation, a port cap's worn to apply pressure and train the skin. And that's seen there at the top left. And this is of course removed for attachment of the giving set for infusion, and you can see the infusion shown next to the port there at the top left. So that just explains a bit about the procedure to implant the device. The next slide, please, Rebecca. So the content of the presentation is on the primary, secondary, and exploratory endpoints, as well as the learnings that we've taken from the trial. Next slide, please, Rebecca. So the primary objectives of the device, specifically to the device of this study, were the safety and accuracy. The secondary objectives were port stability and patency. And we had an exploratory objective surrounding the examination of the infusion within the brain. Next slide, please, Rebecca. So just a couple of terms here, because I'm going to focus on the adverse events before the first treatment dose with CDNF or placebo. More specifically, those related to the device, which are called adverse device effects. And if they're serious, they're called serious adverse device effects. And you'll hear these referred to as ADEs or SADEs or maybe FADEs. Next slide, please. So before the first treatment dose, there were 38 ADEs in total, and it should be noted that 1 or sometimes 2 test infusions were done before the first treatment dose, and this was to make sure that all was okay and to examine the infusate distribution in the first 2 patients. I'll come to this a bit later because that was the exploratory endpoint. So most of the ADEs were mild or moderate, grade 1 or 2, but there was one which was more severe, a grade 3, which was a small bleed in the brain. And these are expected when performing stereotactic surgery and are generally of no consequence. The ADEs are listed here by category called system organ class, the very top level description of the events that can occur in trials. I'm going to look now at the 3 categories with the largest occurrence and break them down into a preferred term, which is a more specific category of the event. So next slide, please, Rebecca. So we'll start with the central nervous system disorder category. And within this, the most prevalent preferred term category of ADE that occurred here was the appearance of a small air bubble, which Magnus mentioned earlier. Now, these were about 2 millimeters maximum in diameter. And there were 8 of these before the first treatment, but they did further on in the trial also. They were transient and they went away over time, and they were safe, and the patients felt no symptoms from them. And they were actually an imaging finding later on in the study, which led to earlier images being examined. They're expected and similar to what can occur in other brain procedures, and they're assessed by the Data Safety Management Board. They oversee safety of the trial, who deem them safe based on the anomalies being small, transient, and, as I said, similar to those sometimes seen in other types of procedure. The next slide, please, Rebecca. So now we move on to the general disorders administration site conditions category, and there were 15 of these, and they related to port site reactions, which are expected as the port site heals. And we've learned a lot about these reactions during the trial, and I'll come to this later on in the learning of the presentation. Next slide, please, Rebecca. So moving on to injury, poisoning and procedural complication category. These were nearly all small bleeds in the brain, and as I said, were expected in stereotactic brain surgery, which I mentioned before. And these were all mild or moderate and had no impact on the patient, apart from one which was more severe. It resolved, but it was mentioned as a possible factor in a subsequent SADE, and I'll mention that in the next slide. Next slide, please, Rebecca. So moving on now to SADEs, and this pie chart describes the SADEs for the whole trial, not just before the first treatment dose, of which there were 6. And to mention that the SADEs, confusion and infection around the port, are part of the same SADE and report from the same form. The infection around the port was determined related to the confusions that occurred there, and along with possible effects from anti-Parkinson's medication. But it was also considered possible that the small bleed I mentioned on the previous slide could have impacted here. The 2 skin tissue decay SADs, I'll come to later on. But there was also one where a patient slipped on a wet floor that was considered to have possible connection with the device as well. The next slide. So this slide just tries to give an idea about whereabouts the SAD occurred during the trial. The healing period shown here is defined as a pause between the implantation and the first testing to allow enough time for some healing before infusion, and was defined as 3 to 5 weeks. Could somebody noise? Maybe the presenters has their microphone on. All SADs did recover, however, and you can see here that I've put confusion and infection on the left-hand side in the same box at the beginning, because, as I said, they were reported on the same form and very much linked. The skin breakdown refers to the skin tissue decay, and the IMP first dose is the first treatment dose. So next slide, please. So another primary endpoint, as well as the safety, as I mentioned, was accuracy, and this was measured in 2 ways. Anatomical, meaning was it within the putamen, which was the target brain structure in the trial, and positional accuracy, which means was it within 3 millimeters of where the catheter tip was planned to be. And remember, I talked about this planning on the second slide. So all catheters were in the putamen, which was good, great results. However, 2 catheter tips in 2 patients were greater than 3 millimeters from the planned target. And we did investigate these, and it showed that 2 of these were due to possible issues with the stereotactic equipment and not part of the Renishaw drug delivery device. And the other 2 were not pushed home properly at the skull, so were slightly not deep enough into the brain. And I'll touch on this in the learning from the trial section in a couple of slides on. And there were also some potential imaging problems with this, which means that the image didn't show quite the end of the catheter tip, and therefore may have seemed a bit shallower. But that's just a possibility. So next slide, please. So moving on to our secondary endpoints of patency, and patency is the absence or the absence of obstruction, if you like. Is it not blocked? Does it flow? And port stability. So no lines became permanently blocked, and in the 3 that did, you can have pump error or user error implicated. And of course, no ports required further surgery for stabilization, which was also very good, and we were pleased with that. Next slide, please.
Jim and audience, I apologize for this background noise. We're trying to get back. We don't currently see any of the panelists or participants having mics open, so there's something possibly wrong with the system here. Jim, I'm really sorry about this, but let's try to continue. And a reminder to everyone in the audience to check that their mics are off and muted.
Okay, the sound seems to have gone now, so that's done the trick, Henry. Thank you. So here is a graphical image of the distribution of the infusate within the brain, and only the first two patients had this analysis. And in these images that you can see here, the purple is the putamen, which is the target, deep in the brain, around 2 centimeters long, with a volume 3 to 5 centimeters cube. And the distribution of the infusate here are shown in green and yellow, and you can see a good coverage of the putamen there in all those cases. So that was good data to have, and that looked good. So I'm sorry, I'm not sure. Yeah, I'm sorry about that. I think the wrong slide is being shown there, my mistake. So as I said, the putamen shown here in purple, with the infusate shown in green and yellow. And you can see there that the green and yellow do overlap the purple, which is a good distribution in the target organ. So next slide, please. So I'm going to move on now to some learnings from the trial. And the trial was very onerous for both the patients and the site staff in the trial. And like Magnus said earlier, our heartfelt gratitude does go out to both of these groups for their help in these learnings and the better understanding that it gave the device for applications going forward. So thank you very much. So the trial helped us get an estimate of the healing period, and by this I don't mean the time between implantation and first dosing, but rather how long it takes for the port to settle down and show less reaction. Reaction around the port site was measured by an assessment called Holger grade, which was done at each visit, along with a photograph also that was taken to capture. Grade 1 and 2 Holgers are considered acceptable, and the graph here on the right, it has all of the patient's Holger grades added up at a particular time point, shown in weeks since surgery. And so you can see how it gets better over time with the Holger scores coming down. And this estimation will get better as we get more data from our other trials, and especially as some of the improvements that we've made are realized. But it was estimated at an average of about 11 weeks. So next slide, please. Another improvement that we've made, and we'll use this in future trials, it wasn't introduced for this trial, is the introduction of a priming aid. Now, those air bubbles that I talked about in the previous slides were safe, but one of the possible reasons for them is that the giving set is not particularly easy to see when it's primed with infusate and all the air has been pushed out by the infusate. And this is important because otherwise that air could end up in the brain. And this priming aid device helps the surgeon see that the liquid is present at the tip of the giving set and hence lessen the infusion of that small amount of air into the brain. So it's a good little device. So next slide, please. So during the trial, we did introduce this port rate. It's a very simple solution. There was general feedback from the sites on how it was hard to keep the hair out of the way from the port sites during the connection of the giving set. And also, one of the SADEs of brain abscess was due to a hair becoming entangled with the giving set and ultimately ended up entrained through the port. And this led to the infection as the infusate flowed to the brain. So the introduction of this port drape reduces the risk of that. And the drape also helps the area, helps make it cleaner and generally easier to attach the giving set. So we had some good feedback on it, and I think it's a good addition to the device. So next slide, please. So for future trials, process guidance software will be introduced that helps the surgeon with the surgery workflow. And there's also been good improvements in the instrument set. They've been designed so the robot moves to the correct place rather than having to adjust the actual instrument itself as to how deep it will go. So it makes it a lot easier to use. And also there's a reduction in the number of those instruments as well. And again, that ultimately helps it make the device easier to use and improve usability. So next slide, please. So we've also updated the training manual for the device and system. So it now has a separate port care chapter for cleaning before infusion, which has been in this trial. And the port site reactions really helped us to develop this, and we learned so much in this trial. The training manual also now has case studies on previous use of the device, and so helps the surgeon, an experienced surgeon, with its use and how to plan and implant the device more effectively. And there's also better guidance on port positioning after blood vessels were cut during the implantation, which led to the SAD of skin tissue decay. And during the trial, we also learned the importance of having a clinical microbiologist or infectious disease specialist due to the port site reactions, and they were very useful. So next slide, please. So we've also updated the instructions for use to include what to do if the port site seems excessively swollen when it comes to infusion, and clarified the cleaning instruction before the giving set is attached. This was following the second brain abscess. The port cap wearing time has been reduced as well. Remember that port cap that I mentioned there on the second slide? It applies pressure, and also following the SAD of the skin tissue decay, where as well as the blood vessels being cut, the port cap and the pressure it applies, it could have reduced the blood supply as well to make the problem worse. And so we changed the amount of time the cap was due to be worn to 12 hours on and 12 hours off. The port caps are changed now each month too, just to make sure they're as clean as absolutely possible. And finally, following the slight inaccuracy of the two catheters not being deep enough, the instructions now make it very clear about pushing that catheter home. So next slide, please. So just in conclusion, the outcome of the trial proved the device is safe and hit the target. And we took learnings from a single center, single surgeon study using a device that was very similar to this one in the past. It was mentioned before, the GGNF trial. And we put it into multiple centers, and having identified improvements, we've implemented them already, and we're pleased with the outcome. So next slide. So it's just to say thank you for your time, and please put any questions into the chat function.